Histology. Something I have been avoiding for 2 straight weeks, but you guys should be lucky I had a really good teacher in my medical school. So I'm gonna be, as always, try to explain in simple students' term without going any straight too far or without directly jumping into any conclusion. I will first explain the basics, then I will improve on that so we can ultimately understand the more complex situations. Now, chapter number 5, Histology, Epithelia. In this chapter, we will divide this chapter in two different parts. First and foremost, we will learn about the epithelial cells and we will learn about the different kinds of epithelial cell, and the rest will be in a second separate video, because if I do it in one video it would probably be 1 hour long. So we don't want to make it that stressful for the students. That is why we will be dividing this in two separate videos. This is part 1 of the two videos, and in this video we will be discussing about the epithelial cell, where they are, how they work, the different parts of it, and all the different kinds of epithelial cells and where you can find them. We will just discuss about where you can find them as a function in just the basal surface, just the basic surfaces, because we will look in them into a deeper meaning in the next video once you have a general broad idea what's going on. So without any further ado, let's just jump straight into it. So before we even discuss anything else, we need to know what an epithelial cell is. Now think of it as you have a ground and that ground is an organ. You can have anything or everything in that ground you want. That's not the concern right now. We have that ground and we need to cover that ground. So basically, normally anyone would just build walls around that ground. Now that wall, think of those walls as a layering, protecting, covering, layering of any organ system. Now those walls are what we call as epithelial cells. So epithelial cells are the ones that line the organ system. So anything around the organ system, epithelial cells, they cover it, and it's just not one dimensional. Think of it as epithelial cells are walls. Like I said, a wall also has two sides. So basically epithelial cells will also have two sides. I will tell you why. Because we need to transport different kinds of structures, and let's just say oxygen, glucose and stuff like that, we need to transfer in and out of the organs. So we need the epithelial cells to have channels which can help transport different kinds of stuff in and out of the organs. So a wall has two sides, so does an epithelial cell. Now let's see which side is where. Both sides have name, but of course I will try to make it as easy for you. One is called an apical and the other one is called the basal surface. Now I'll let you to decide which one is on the bottom, which is stuck to the organ, which one is on the base. Simple, the one which have base in its name, the basolateral surface, is the one that's gonna be in the bottom side and stuck to the organ. The one above it, the one that is facing the outside cavity, the outside lumen cavity, that is the one, that side is what we call as the apical surface. The base which is stuck to the organ, the base is the basolateral, and the top one is the apical surface. Now like I said before, we need to transfer stuff in and out of the organ and through the epithelial cells. For that to work, the epithelial cells need to be polarized. Now what does that mean? It means that both surfaces must work differently. It both are polarized, they both have different structure, different ways of working, and that is how we allow all kinds of stuff to pass in and out of them. Now as we know already, epithelial cells need to transport stuff in and out from them. There are actually two ways which we can do that. Number one is transcellular and number two is paracellular. Now Transcellular, think of it as a transport. So big molecules, we are transferring it through the epithelial cell. Transcellular, transfer, transcellular, we need something or someone to transfer it, and those are the epithelial cells. So transcellular will let those molecules pass through them. So transcellular is when they pass through the channels of epithelial cells. And the remaining one, the other one, paracellular. Paracellular is when we let those molecular pass in between epithelial cells. So let's say you have an epithelial wall, this is one epithelial cell, this is another, but paracellular is when the molecules transfer between two cells. So this gap where my face is, this is where the channel, the gap, this is the gap where the molecules will pass through in between two epithelial cells. So let me give you an example. Let's say it's either in kidney or intestine or anywhere else. Let's say we need to transfer stuff in between them. So how would it work? Of course, the epithelial cells are polarized, so there are two, apical and basolateral surface. So basically, let's say we are trying to pass chlorine molecule. So the chlorine molecule will come through the apical surface, they will pass through the apical surface, and then they will pass through the basolateral surface, and that will create a ionized polarity. Since chloride ions are negatively charged, they will attract sodium molecule. So we already have chloride molecule, which is K. Now we already have chloride molecule, which is Cl. Now we will also attract sodium molecules, which is Na. So Na follows Cl. Why? Because Cl is negatively charged and Na is positively charged. So Na will follow Cl. And now what do we have? We have sodium chloride, which is salt. Now, as I have discussed in previous video, hypotonic solution, hypertonic solution. Hypotonic solution is when we have excess of water, less salt, and hyper solution is when we have too much salt and less water. So they will want to be in the middle where the equal amount of salt and water are present, which is the isotonic solution. So now we had chloride molecule and then sodium followed it. So now we have NaCl, we have salt in the cells already, but this has now made an excess amount of salt, which is a hypertonic solution. So as naturally water will follow through after the salt to maintain the isotonic solution. We don't need extra salts, we need the equal amount. So when salts pass through, after that the water will follow, and then we will have a balance. Now that we know about this, let's talk about a clinical case. Cystic fibrosis. Cystic fibrosis is one of the most common hereditary problem. In this problem, we just discussed about the chlorine molecules. Let's just say the chlorine channel, the channels which the chlorine is passing through, let's just say the chlorine channel do not work. It is a genetic hereditary disease. If the chlorine molecule channels does not work, then the chlorine won't be able to pass through, and when the chlorine doesn't pass through, we won't have a salt, and as a result the water will not follow because we don't have any salt. So to the balance the salt, we won't have any water to follow through. So as a result, what happens in the lining of lungs, intestine, kidneys, etc., now that we do not have water, the mucus lining of those cells will just be a thick mucus. The outer lining will just be a thick mucus without water, and that thick mucus is caused due to the inefficient working of the chloride channel, and this condition is what we refer to as cystic fibrosis. It affects the lung, the intestine, and all different kinds of the body. It is an hereditary disease, like I said before, and the cure to this, as per to my knowledge, has not been found yet. So we already talked about the transfer through the epithelial cells, but what about the existence, the survival of the cell itself? Of course, you would think for the cells to survive, we would need nucleus, cytoplasm and all different kinds of stuff for the cell to survive. But those cells does not have any blood vessels in them. So as a result, the epithelial cells, they do not have any blood vessels of their own, and you will always find these epithelial cells right next to some blood vessels where they can transfer the oxygen and the nutrients from the blood vessels to them. So they do not have a blood vessels of their own, but you will find them always right next to one. Now we have discussed the first part of this video already. Now let's discuss about the types of epithelium. Basically, we can divide the epithelium into two different parts: simple epithelium and stratified epithelium. Now you would think, what is the difference between simple and stratified? Let me tell you. If you have one cell, let's say basolateral surface and apical surface. Apical surface is facing the lumen. What is lumen? Lumen is the tubular cavity of the organ outside. The tubular cavity, lumen, that's the outside, and the basolateral surface. So between basolateral surface and apical surface, if we have one single cellular layer, then that layer is simple, as it means single is simple. And if we have, let's say that's the basolateral, that's the apical, but we have multiple layers in between them, then those cells are the stratified. So stratified are multiple and simple is simple, just one. So let's move on to the different kinds of cell. Number one is simple cuboidal epithelium. So all this, we use the H and E chain, as you already discussed. You already know about the H and E chain in like the primary standard. So the H and E chains helps identify the nucleus as blue and the epithelium as pink. They color it like that. But let's say the L right here, we don't focus on this white part. This white part, which is L, is the lumen. The lumen I talked about, the one that's apical surface is facing, yes, that is the lumen. And the arrow right here shows the epithelium surface. So we need to focus on that. Let's focus on this. So simple cuboidal. Simple means there's only one layer. So let's see. We have the lumen here and we have the cells here. So as you can see, there's only one layer. Anywhere if you see, there's only one layer, which makes this a simple. Now cuboidal would mean it has the same width and the same height. So let's focus on this arrow right here. This has the same width and the same height. So it is one layer, which makes it the simple, and it has the same height and same width, which makes it the simple cuboidal kind of epithelium. Now where would we find it? So we don't need to memorize all the things. What we need to do is just figure out what kinds of cell they are and their function, which I will tell you in few minutes. So you can just decide which epithelial cell would suit where based on their functions, which would make it even more easier for you to recognize which ones were, and you don't have to memorize anything. So you would find this simple cuboidal epithelium in the renal tubule, or you can find them in the salivary gland, both the simple and stratified. Keep that in mind. Simple and stratified cuboidal, you will find them in the salivary gland. Now that we know what is simple cuboidal epithelium and where it is, let's move on to the second one. Now the second one is simple columnar epithelium. As the name suggests, column, pillars, column. Simple, it would be just one layer, and column is like a pillar, so it would have less width but a much larger height. So a simple columnar epithelium. Let's discuss this. So as you can see, the white part here is the lumen, and right here you see This is the nucleus and this is the cytoplasm. This is the epithelium cell. As you can see, it's not just cut off from here. So the width and the height are not the same. The width is like this, just this much, but as you can see here, the height is much larger than the one we had in simple cuboidal. So it's like a pillar, like I said, a pillar or column. The name itself suggests the shape. So simple columnar, we have this much width and that much height, much more height as compared to the width, which makes it like a pillar and like a column. So simple columnar. Why simple? Because the lumen to the outside, it's still just one cell. Do you see any extra nucleuses behind it? No. Why? Because it's still a simple epithelium, so it's just one layer. Now you will find this epithelium in the GI tracts, usually the small and large intestine. Alright, so this third one is an interesting one. This is the simple squamous epithelium. Now let me tell you what squamous means before I even explain in anything else. Simple squamous epithelium. Squamous just means flat. It would just be flat and very thin. So this green arrow, it indicates the white lining of lumen, and facing that white lining is the epithelial. It is so thin, it is tiny, it is thin, it is squamous. That's what it means. It's tiny, it's thin. So this green arrow suggests the lumen facing part. Hang on, let me zoom it in. Yes, so this lining is the simple squamous epithelium. Now as you can see, the white lumen here, the white lumen here, but still there's only one cell. You can see my cursor, there's still only one cell between lumens, so it still indicates it is a simple, it's a squamous epithelium. It is very thin, it is a squamous epithelium. So but it still have one layer between lumens, so it is still a simple squamous epithelium. Now one of the most important thing to remember about this one is, since it is so thin, why is it so thin? It is so thin that you can easily have things pass through it. So remember I said you just need to know the functions of them and then you can figure out everything. So it is so thin that it is easier to pass substances through them. So this kind of cell will be in areas where you will need to have exchange of stuff. So let's just say lungs. It would be in the alveoli where we need to exchange carbon dioxide and the oxygen between the alveoli and the blood. So this simple squamous epithelium, we will find it there. In the nephron, in the kidneys nephron, where we need to have high exchanges of waste material through the water, and we will find this there because we need to have a high exchange rate. We have more surface, it is flat, we need to have more exchanges. So you will find this kind of cell in the lungs, in the nephron and stuff like that. So remember, you don't have to memorize it. Now you know its function, now you know its shape, now you know its function, so it's easy to figure out where you will find it. So the next one can get a little tricky. This, you might think it is a stratified, and since it is long, it is a stratified columnar epithelium, but that is not the case. This looks like a stratified epithelium, but it actually is not. Okay, so follow my cursor. You see this epithelium, it is attached to the base. Okay, fair enough. Now let's talk about this one, the one above it. Oh, there's two layers, so it might be stratified. Wrong. You see this epithelial cell, this one is also connected to the basolateral surface. Oh, okay. So what about the one, let's just say in the third row, let's just say, what about this? Is this stratified? It might be. It looks like it's stratified, but it is not. As you can see, it goes through here, that's the nucleus, but it just goes through here and through all different kinds of epithelium, it still touches the basolateral surface. So it looks like stratified, but it is not. So that is why the name pseudo stratified epithelium. It looks like it's stratified, but in reality it is not. It is still a simple, but it looks like stratified, so we will name it pseudo stratified because it still has multiple layers. Now if you focused on this part, you can probably figure out where this is. You can find this cell in trachea and epididymis. As you can see, you can probably figure out. If not, do not worry, do not stretch out, nothing to worry about. It is just found in trachea and epididymis. Oh, now this is the one I am excited to tell you about. This is neither a simple one nor a stratified one. It might look like a stratified one because it's complicated, but it is not. This right here is a special kind of epithelium. Now, you may think about what's so special about this. It just looks like normal to me, but let me tell you this. This is the transitional epithelium, urothelium, which you can also refer to as the urothelium. This cell right here, as the name suggests, transitional epithelium. What's so special about this? This epithelium can shift its ways. So if you can see, we have, let's just count how many epitheliums we have on top of each other. So we have 1 2 3 4 5 6 7 8. Let's say we have 9 layers right now. But before I explain it to you, let me tell you where it is, which would help you understand better on why this is a special kind of epithelium. Now, this kind of epithelium is usually found in places like the ureter or your bladder. Now, that usually is this part right here. This part, the bladder is empty right now. Let's just say when the bladder fills up, this epithelium will shift its way. So right now we have 9 layers, but when the bladder is full, it will stretch out, it will transition. See what I did there? Transition from 9 layers when it stretches out to 2 or 3 layers. So it transitions from 9 to 2-3 layers, and it is almost impermeable because you already did all the work, all the effort of filtering out everything. Now all you have is the waste product, the urine in the urethral bladder. You do not need it to be diffused out of it. So you need it to just go through and not diffuse. So this is impermeable. This shifts its width, but it is impermeable. So in the urinary bladder, it will go from 9 to 3 when it's full, and then when you empty it, it goes back to 9 or whatever. This is what makes it the special kind of epithelium, the transitional epithelium. Now let's just go to the second kind of epithelium. We just discussed about the simple ones. Now let's just discuss about the stratified ones. Oh, this might seem scary, but worry not, there is nothing to be afraid of. Let me explain it to you. Let me break it down to you bit by bit. So as you can see, there are like, I'm not even gonna count, there are way too many layers on top of each other. These are of course the stratified kinds of cell, but let's see the width and the height. Now let's talk about what this is. This is a microscopic image of the skin, the epithelial cells. Now these are the thin kind of epithelium. Thin, what was it? The squamous epithelium. Now it is on top of each other, so it is the stratified squamous epithelium. But let's just say what's so special about it. To understand this top layer, let's just know where we will find the stratified squamous epithelium. You will find two types of this, keratinized and non-keratinized, in either the esophagus or on your skin. Now keratinized or non-keratinized. What does keratinized mean? Keratinized means the hardening of it. Your nails, your hair, that is keratin. So these nails are keratinized, which would increase this top layer, which would mean the top layer is the keratinized cell. Now it is hardened, which indicates where this is. Is this either esophagus or the skin? Now this kind of epithelium, the stratified epithelium, you will find where you will have lots of abrasion as in lots of rubbing, lots of eroding. So this can multiply easily and just go on top of each other and replenish it. Now this keratinized, why do we have this? The keratinized is the dead cells, the dead squamous epithelial cells. These ones are the live ones, but this melted layer, this is the keratinized, the dead cells. Why do we have that on our skin? Why do we have dead cells on our skin? The answer is simple. As I discussed about the squamous, they are very thin, so it is very easy to transfer things between them. Now our body, we need water to stay alive, and since it is so easy, we can just have water pour out of us from these cells. So to prevent that, we make this keratinized layer, we have it on top of the live ones to help stop, prevent the water go from our body to the outside. So to prevent the loss of water, we have the keratinized cells on top of the live ones. Now the second kind of stratified, they have the same width, they have the same height, but this time they are on top of each other. So we have the stratified, but we have the cuboidal epithelium. This white is the lumen. The cells are cuboidal, but now they are stacked on top of each other. They are stratified. Remember what I said, both simple and the stratified. Where would you find them? In your salivary glands. They are usually found in ducts and salivary glands. So the ducts, they help secrete. These are the cells which specialize in the ducts. You don't have to go too deep into it. We will go through that in further chapters. Do not worry about it in the next few videos. That's all you have to know about this chapter right now in this video. So let me recall everything for you. Epithelial cells are on top of organs, they cover it. They have two layers. The top one is the apical layer, and the base one, the bottom one, is the basolateral surface. They are polarized, so they are both different. None of them are the same. They are both different, which helps the molecules to pass through the epithelial cells. Molecule can pass through two different ways. Transcellular transfer through the cells, or paracellular, in between cells. Now we can transfer chlorine, sodium will follow, then we have salt, and to balance that, we have water follow through it, which makes the balance. But if the chloride channels aren't working, then the water won't follow, and in result we will have a thick mucus layer, which is a hereditary disease who we refer to as cystic fibrosis. Now epithelial cells do not have blood vessels of their own, which is why you will always find them right next to any other blood vessel, and they just transfer oxygen and all the molecules from those blood vessels to themselves. Epithelial are divided into two different kinds of epithelial: simple and stratified. Simple is one layered, stratified is multiple layered. Simple layered has simple cuboidal, width and the height are same. Simple and stratified, both you will find them in the salivary glands and stuff like that. So simple cuboidal is in your salivary gland. Simple columnar, we have column like pillars, you will find them in the intestine. And simple squamous means the flat one, so you will find them where you need to have more transfer of stuff. So you will find them in your lungs, your alveoli, your your nephrons in the kidney and stuff like that. We have a special kind of epithelium which is transitional epithelium. You will find them in your bladder. Transitional, as the name suggests, they transition from having thick 9 layers to 2 to 3 layers thin when the bladder is filled, and then they can shift back to their original pacing. Now they do not leak any molecules through, so that is why your bladder is safe. The urine in your bladder is not going anywhere, don't worry. Now discussing about the stratified one, we have different kind of stratifieds. Let's just talk about the first, cuboidal stratified. In the salivary gland, same one, same height, same width, but they are on top of each other. You will usually find them in the ducts. Now the other one is simple squamous epithelium. Squamous is the flat one, which flat, so you will either find it in your esophagus or on your skin. So stratified squamous, but since squamous are so permeable, they just let everything pass through, so you will have water loss as a result from your skin to the outside. So you don't want that. That is why we have keratinized squamous cells on your skin. So the dead layer of stratified squamous cell, they cover the live layer, which helps prevent the water loss from your body. This is all you needed to know. This is the first part of the 5th chapter. It sounded complicated, but when we broke it down to different levels, we started from the basic. Now we have a clear figure of what we needed to know, but now you don't have to worry about it because you know the basics, and which will help you understand the complicated stuff in the next video. So this is all in this video. I hope it was easy for you. As always, if you have any questions, you can write them in the comment below, and I will, as to the best of my ability, respond to you as soon as I can. I hope I notice it, but this is all. I hope you guys liked it, and I will see you guys in the next video. Goodbye.